Today to have with us Dr. Chandrabali Bhattacharya. She's an assistant professor at the Department of Chemistry and Biochemistry, part of the mRNA nanomedicine program. It's a interdisciplinary biomedical science and engineering program at UNLV, otherwise known as University of Nevada, Las Vegas. And the title of her talk today is Endogenous Targeting Lipid Nanoparticles, or ENDO, platform for systemic delivery of mRNA to the pancreas. Very cutting edge and hot topic these days. Just a little bit about her, a short bio. She did complete her PhD in bioorganic chemistry at ASU, studying carbohydrates and with tumor targeting properties, and then spending a short period at Caltech examining the role of heparin sulfate glycosaminoglycans in neuronal development. She joined the Koch Institute for Integrative Cancer Research at MIT to complete her postdoc under Robert Langer and Dan Anderson at the Koch Institute. At MIT, she worked on the development of novel class of supramolecular structures with promising glucose binding properties for the development of smart insulin systems and engineered lipid nanoparticle platforms, or LNPs, for in vivo delivery of CAR mRNA to T cells, advancing adoptive T cell cancer therapy. Currently, her lab integrates small molecule chemistry, biomaterial design, immune engineering, and regenerative medicine to engineer next generation LNP platforms for precise organ and cell targeting. And her achievements have been recognized through several awards recently, including JDRF Postdoctoral Fellowship, JDRF Advanced Postdoctoral Fellowship with a Faculty Transition Award, and the Pharma Foundation Starter Grant and Drug Delivery Award, and most recently, excitingly, an NIH R35 MIR Award. So congratulations on all those accolades. That's amazing. And we are really excited to hear more about your talk. I'm going to ask people to drop questions in the chat and she'll answer at the end. And there's also a link to her recent paper there.
Thank you for your introduction. So as you said, our lab mainly focuses on developing lipid nanoparticle platforms that can reach that is specifically designed to hard to reach organs and cells. We know the central dogma of biology: DNA produces RNA, RNA produces protein, and literally every disease is related to either upregulation or downregulation of the protein. And mRNA can be used to upregulate protein, or siRNA can be used to downregulate protein. So basically, RNA can be used. Can be potentially used for different kinds of treatments. Nearly for every disease, we can find an RNA treatment. RNA treatment. But turning RNA into drug is really hard because RNA is unstable. It has positive, negative charge, so it cannot cross the cell membrane, and it doesn't know where to go. So it needs a delivery system to do its function and produce the protein or decrease the production of protein. So LNPs has been known for a while to deliver mRNA to different locations and produce protein. And we know COVID vaccine, how it was revolutionized the treatment and help people. And so our research is more focused on targeted delivery, if we can modulate these nanoparticles for different organs, which is again hard because they will, So, but these are very useful because they can result in situ production and systemic secretion. So your body can act as a drug depot and release these drugs over time, which is a very nice thing. So what are the basic components of nanoparticles? Lipid nanoparticles are made up of mainly 4 components: ionizable lipid, phospholipid, cholesterol, and PEG. Ionizable lipid is, as from the name, is ionizes at lower pH, and at neutral physiological pH, it's not charged, and phospholipid, it like complexes with RNA. So ionizable lipid complexes with RNA. Phospholipid, it forms the bilayer structure, and the cholesterol is, as we know in biology as well. So it's the part of the membrane, right? So cell membranes, they also have cholesterol. So this provides rigidity to the LNPs. And then PEG is there to increase the circulation times and provide stability so that they don't march together and increase circulation time. Yeah. And they are mixed, these components are mixed together to give these nanoparticles, which are like 50 to 100, 150 nanometers in size, which can carry RNA. So one of the example of how these structures look like is for COVID vaccine, Moderna COVID vaccine, and the components, they look like this. So the ionizable lipid is SM-102, and it's a branch lipid, and then phospholipid DSPC, cholesterol, and PEG. So there are, other than SM-102, there are two more ionizable lipid which are FDA approved. One of them is for the BioNTech vaccine, ALC 0315, and MC3, which is a component of Npatro, which is the first mRNA drug, which is an siRNA drug ever approved by FDA. So how do they work? So when something is injected in the blood, these LNPs, they travel to the blood and they reach the cells. So once they reach the cells, there are receptors. So they interact with these receptors, and with the help of the receptor-mediated endocytosis, they go inside and they reach the endosome, where they get protonated. The ionizable lipid get protonated, and then they break and they release the RNA. And this is called endosomal escape, and then they produce the protein. So it sounds pretty much simple, but the main problem, what is the major problem in the field, is when these LNPs are injected in the blood, there is a protein called apolipoprotein E, which codes the RNA in the blood. Which codes these nanoparticles, and then they traffic these nanoparticles directly to the liver. And because they hijack the body's mechanism of cholesterol transport. So basically, that is like different kinds of LNPs injected in blood goes to the liver. So our research is how, and there are several other groups who are trying to brainstorm, how can we stop this coating? How can we stop this interaction of these LNPs with ApoE and redirect to the different organs? So one of the way to do this is to add a fifth component in the LNP, which was shown by Daniel Siegwart back in 2020. And he showed that when we add cationic lipids to the mix, it kind of changes the surface decoration and interacts with different protein, and it goes to the lung. When it is added, a negatively charged lipid is added to the mix, it then coats the surface in a different way, and then it interacts with something else, and it goes to the spleen. And then you can see that there is expression for when it is DOTAB, which is a positively charged lipid, it goes to the lung, and we see very strong lung expression. And then the corona study revealed that vitronectin is a protein in the blood which is responsible for this. Then when it was coated with negatively charged, when the negatively charged component is added, it goes to the spleen, very strong spleen expression, and beta-2-GPI is another protein which is responsible for this. So based on all this, our approach was, can we use a biologically active ligand which is already present in our body and do the same kind of redirection? Maybe they will interact with the receptors and hijack some other pathways in the body, and we can reach different targets. So what we did was high throughput screening, because it is very hard to pinpoint the decoration or the composition which can lead to such kind of targeting. So we we used a fifth component as well as high throughput and technique. So we created 100 nanoparticles. So we have 5 different ionizable lipid, 5 different vitamins, and then four formulation, and we had 100 LMPs. And the reason for using vitamins that they have shown so much different interactions with different cell types, and especially we were interested in vitamin D, which has huge role in different kinds of cell growth and especially eyelids. So we were very excited about that. And then after we got these nanoparticles, we did a high throughput analysis, which is called batch analysis, because it will be crazy to, one by one inject 100 nanoparticles in in the in the mice. So like so they yeah. So what's this in like batch analysis is, so as you can see there are the different combinations we chose, the batches are those combinations. So first we used formulation ratio. We batched all the nanoparticles based on formulation, then we batched them according to ionizable lipid and then endogenous vitamins. So what does that mean is we had 100 LNPs. Then we batched them into 4 different formulations. So each batch will contain 25 nanoparticle, and we intravenously injected them. And to our surprise, we saw formulation F2 and F3 had no liver, almost very less for F3, but there is nothing going to the liver, so which was pretty exciting. And then we saw a huge signal in the pancreas, which was I've never seen that, so it was very exciting. And the quantification also gave us more excitement because it was pretty much higher. You can see how much it is higher than MC3 in the liver. So, which is MC3 is the other FDA approved ionizable lipid I was talking about. Now next we took those 2 batches, F2 and F3, and then we pooled them together. So it is more, we can call it split and pool. So we pooled them together, and now we have 50 LNPs. We batched them in 10 different, so five batches, 10 LNPs per batch. And we saw this ionizable lipid C12-200. It showed very strong pancreas signal, and there was nothing in anywhere else. So that was very promising. And it was almost 10 to the power 8 value, which is a very good value for our field. The next thing we wanted to see whether these SC12, then next thing we batched these C12-200 LNPs, which were 10 LNPs into 5 batches, because these were vitamins. And we saw a cholecalciferol, which is vitamin D, is still holding on strong, and it shows the pancreas signaling. So now it also shows a similar 10 to the power 8 signal. And but it is a mixture of 2, so we individually validated both the formulations and compared with C200, and we saw both the formulation F2 and F3 had pancreas targeting, and it actually very, very surprisingly, there was 99% redirection from the liver. So there was 99%. So C12-200, why I'm comparing with this, is the traditional formulation, 4-component formulation. And then when we added 5th component in C12-200, from 99% liver accumulation, it redirected to 99% pancreas accumulation. So which was very promising. And then we wanted to test this, whether these LNPs work in other animal models, especially in AI14 transgenic mice model, because this is a gold standard for us, for our fields, where you can understand cell type. Why? Because this has a cassette, LOXP, stop, LOXP, TD tomato. And if you have delivered, if a cell contains, if its cell receives this Cre mRNA, it produces the Cre protein, and then it can cut this LOXP stop out and can produce this tomato, and it's very bright and hard to miss and good analysis. So we saw similar data that there is very high production of TD tomato and very less in a liver. We have a liver image in the supplemental. If somebody is interested, can go back to the manuscript. Yeah, so that was pretty promising. And now we wanted to understand which cell types it is going to. And we used immunofluorescence, and we saw that it is going to pretty much everywhere in pancreas, but it is also very interestingly going to the alpha and beta cells, which is, I think, a very interesting data, and it has huge application in type 1D, and in coming years we'll see what it can do because we are very excited about this. So next we wanted to see whether our platform is what's the toxicity, is it even translatable? So we did a lot of the ALT, AST, liver function, Cre, bun, kidney function, and different kind of cytokine analysis, but which is also in the, I just chose a couple of them, but there's all back in the paper. And we saw mostly that they are comparable to PBS control, that background. And C12, you can see C12-200 is pretty much it is very toxic compared to our CQF3. And we also have compared CQF3 with MC3, which is also in the supplemental data of this manuscript, and it has shown similar FDA-approved MC3 is FDA-approved, so it has similar toxicity. And I believe this reduction of toxicity is because you're substituting these ionizable lipid, which are potentially the cause of the toxicity for LNPs. With a huge chunk of that is replaced by this endogenous vitamins, which are non-toxic and are in the body. So next we also did some histology, and we saw there is no difference between the PBS and CQF3. And then we also checked the body weight, and you could see T12-200 has some changes in the body weight, but the CQF3 is almost comparable to PBS. So based on this, we concluded that it has pretty safe, minimal toxicity, and has a potential of a repeat dosing. We haven't done the study yet, but that's in future. We want to test if we repeatedly dose people with this, like animals with this, can it be, how much is the tolerance, which is another huge problem in the field, especially if this can be used as a type 1D medication, or especially replacement of the beta cells. So anything, it will be like it will need multiple dose. So now the main question which comes into mind, how this is very strange data, right? It is going directly you're injecting into the pancreas in the blood, and it's going to pancreas selectively. And how is it doing that? Which is like the selectivity was pretty striking. So yeah, so we thought about if it's a vitamin D, so it might be some vitamin D receptor is involved in doing something, right? So vitamin D receptor knockout mouse, they are available, but it's pretty inaccessible. So what we did is we used a traditional technique where you have a receptor, and if you add some small molecule, it get blocked, and then it doesn't function what it's supposed to do. So we found a vitamin D receptor agonist that can block a receptor, and then nanoparticle will not be able to bind those receptor, and then we can see whether our hypothesis is correct or whether VDR is involved. So we found this antagonist METC7 VDR antagonist, and which is well known antagonist. So, and what we did, we treated our mice with METC7, and 12 hours later we injected our LNPs, and we saw that the complete disappearance of the signal. So, which strongly supports that these nanoparticles use VDR to reach the pancreas. So to be more certain, what we did is we replaced the vitamin D with the VDR agonist in our formulation. So if it is, because it's a, yeah, and we wanted to see whether it is doing the same thing. So if our, to further strengthen our story, and and absolutely it is doing the same thing. So when you replace vitamin D with a vitamin D vitamin D, any kind of vitamin D binding small molecule, it is like that is, it should go to the pancreas. So which was pretty exciting, and we saw the similar kind of targeting. We saw similar kind of no difference in signal, like we in total flux, which is similar. And we also saw the similar kind of redirection, 99% to the pancreas. So the next question is, if we inject, so previously we have seen that there is the published data where intraperitoneally was intraperitoneally able to reach the pancreas. So we wanted to see that if our material can reach the pancreas with IP injection, or it is just an IV thing. But to our surprise, we see that both IP and IV injection of CCOL-F3 reaches the pancreas. And it is the amount of the nanoparticle reaching the target probably is lower using IP, because it probably from the IP space it goes to the circulation and then distributes, but the selectivity is still intact and preserved, and it's pretty fascinating. So then we wanted to see whether can we deliver other nucleic acids, because that would be amazing, right? Because we can look into different therapies. Then especially we looked into plasmid DNA and circular RNA. The good thing about circular RNA is because it's circular, so it can be it is stable in the cytosol for a longer period of time. So that kind of helps. Then the amount of dose you're going to give is lower. The frequency of the dose is, you can reduce the frequency of the dose. And also for DNA, you can do one injection, and like that should work for many months, right? So we also tested that, and we saw the same results. CCOL-F3 is a very versatile platform which can deliver pretty much different kinds of nucleic acid, DNA, circular RNA, to the pancreas. And the selectivity is still intact, it's still 99%. And so, which was very exciting. Yeah. So based on all this, we feel that CCOL-F3 is pretty exciting platform, is ENDO platform, which is the endogenous targeting platform, is a very exciting platform, and it has huge potential. And our star LNP CCOL-F3 can deliver different kinds of RNA, and it can be delivered intraperitoneally, intravenously, and uses the VDR receptors to reach the pancreas. We probably need to get more in detail, more in depth how it is doing that. But that isn't, we'll be doing in future. But I think this can help different therapy can help develop different therapies for type 1D, and we are very excited about this. And I'd like to thank my team, Ivan Isaac, who is who's did all this work, and Pharma Foundation, who had helped us in the college with all startup money. We are very grateful for all the support from the department and the institution, and we like to also thank you for your attention. And if you have any questions.
Fantastic. Thank you, Chandra. This is really, an exciting time for your laboratory as you further develop these delivery vehicles for potential therapies. I just want to open up to any questions. Anybody want to ask a question? Either drop it in the chat or unmute yourself. Nothing yet. I had a question. So when you're seeing the targeting to the islets, can you do any post-mortem studies to see exactly which cell types are targeted in the pancreas, sorry. Are you looking at exocrine cells, or islets, endocrine islet cells, or a mix, or what does that look like?
So right now we did only immunofluorescence, which could tell you like a very generic, which can give you a generic view, which we had in this, uh, like, sorry.
Sort of a high level view, yeah.
Yeah, which is a high level view and which you can see that it's going to the islets. But we would have to further dive deeper and see which specific cell types. But from the high level view, we can see it's going everywhere. But
Yeah.
again, we can go. This is a great first step. We can definitely find more, fine tune this platform more to selectively target one cell type in in this mix.
Right, yeah, because you'd like to, right, if you're doing something specific to beta cells, you'd like to target them specifically, et cetera.
Yeah.
And so also I want to ask you, vitamin D, right? It's known to modulate immune responses. So does like its incorporation reduce off-target immune activation, like, or improve tolerability, or
We
in
haven't
any way that
done.
could benefit, sort of the autoimmune in context of autoimmune T1D?
It can probably, because there is so much literature on this, but here it is like on the surface, so it's kind of reaching directly to the cells. But of course, if it's reaching directly to the pancreas and when it gets degraded, the nanoparticle breaks. Of course,
Mhm.
there will be more vitamin D in the cells where it's in the target. So definitely, that is something we would interesting to see. But no, we haven't done anything like that before. We are very early in the development. We have just developed the platform, but
Mhm.
we are interested in taking it farther and see how we can use this for type 1D.
And then how can you talk a little bit about sort of the VDR, like as the cells become sicker, right? And more inflammation, the VDR might change the docking systems available for the endo, um, for your endovesicle might change. So are you thinking about windows of time when this would be best to deliver cargo? I mean, if you're thinking about like maybe it might be best to deliver cargo prior to diagnosis of type 1 diabetes, prior to having the cells, really inflamed and under stress. Have you thought about that sort of windows of opportunity with this kind of platform?
It's actually an excellent question, but I would think that this platform can be fine-tuned to different windows for different applications. For
Yeah.
example, if you probably earlier in the development, pre-diabetic and those kind of
Just with like one or two autoantibodies.
yeah, so then you can use an increased tolerability at that time using this platform. And later in the platform, maybe you can use this to transform some of the other cells into beta cells, which will, because it's pretty much going to other cells as well. So there is a huge, there is beta cell regeneration, tolerability, different kinds of application which can come out of this.
Can you talk a little bit? So we're sort of talking about the progression to type 1 diabetes, the natural history. But can you talk a little bit about islet transplantation, how this system might be used um, in that strategy?
Yeah, so we are also thinking in that direction, but I can't really talk a lot about that.
Not
But
yet.
yes,
Okay.
yes.
Okay. Yeah, because it seems like it has a lot of applicability
Yeah,
to
yeah.
both prevention and potentially rescuing. So yeah, it's really, really exciting times. Are you working closely with the MIT at all with this strategy, or
No, no, we are not.
who are your top collaborators right now? Or are
Uh,
you looking for collaborators?
I am looking for collaborators,
Okay.
but yeah, I'm also yeah, not have started active collaboration, but yeah, we
Yeah.
I'm yeah.
Okay, this is great to broadcast that out there that you're looking for collaborators and you have a really interesting system. So we'll blast it out there and hopefully you'll get some inbound.
Yeah, thank you.
Last,
I'm do working with someone at UMass. We haven't started. Uh, we are in the early phases, and we'll see how that goes.
great. We do have some questions. Alex, do you want to unmute
Yeah,
yourself?
yes, yeah. Yeah, it's nice talk. Yeah, thank you. I'm just wondering like how you choose vitamin D. Yeah. I mean, there are a lot of like different components, molecular you can choose. Why specifically choose vitamin D? And if we want to target other specific organs or like specific cell types, how can we like get the rationale to know like what kind of molecular we should choose?
So, great question. So we didn't just choose vitamin D. We choose 5 different vitamins here.
Mhm.
So we chose them based on their interactions with different. So we wanted to see if these different endogenous vitamins can do can interact with different systems and do different kind of different redirection to different organs. So that was the
But
how we started. But
yeah,
definitely we reach the pancreas, which is very exciting and closer topic to me, that we can use this platform and to selectively target one of the cell of the entire population's targeting at the point. We can definitely add more targeting component to endo and then do that. So it's all in the formulation. So formulation
yeah,
is key. That's why you got to make a lot. You have
yeah,
to screen a lot.
yeah.
Yeah.
Yeah, but why you choose vitamin?
Why did I choose vitamin, or why do I choose
Yeah,
vitamin?
yeah.
Why did I choose vitamin? Because it is endogenous. I wanted to uh, use so it is like nothing really. So I was also interested in looking at immune targeting. So I thought vitamins have their interaction with various immune cells. So
Yeah, but
yeah, so that was how I started to think about it. That
Yeah, it
yeah,
sounds
so
almost as if it was like a serendipitous, right? Because then
yes,
when
it
it
is. I
showed
would
up,
call
you
it that.
started out looking at one thing and then it ended up being a high signal in the pancreas. So that was really,
Yeah,
really
this is serendipitous, but we did include vitamin D. Um, yeah, we were not hoping to go to the pancreas because we have never, never see anything go to pancreas intravenously. So
yeah, is that possible to do IV injection as well?
no, this is
Intravenous.
IV. This is all IV injection.
Okay, yeah, good to know. Thanks.
Thank you very much. Vanessa Hammond, would you like to unmute yourself?
Hi, hi Chandra. Really nice talk. I want to quickly introduce myself. So I'm working here in RNA delivery at Bayer, and in the last weeks I looked very deeply into your papers. It's really impressive. Also your latest paper on the cortico LNPs. So yeah, I have a couple of questions actually. So my first one would be about the uptake mechanism. you state that that the VDR is responsible for that, but to my knowledge, it's basically an intracellular receptor or more like a transcription factor. How do you explain this?
Yeah, so I did explain that in the paper. So VDR is also known to be present in cytoplasm as well as on the surface. So it is mostly a nuclear protein, but it can shuttle between places. So we believe that is so, and as you are right, that VDR is mostly present everywhere. So I believe that because of this, how much VDR is on the surface, it is going to the pancreas. Maybe that is next step. We need to see that maybe there is more VDR in the pancreas on the surface. That's why it was taken up by the pancreas, not other organs. We even checked bone and stuff. Bone has a lot of vitamin D known to have VDRs and vitamin D. So there was nothing in the bone. So we didn't put it in here, but yeah.
And do you think that the vitamin D in the formulation basically then changes the protein corona so much, like that we have coating with the vitamin D binding protein, and that then this leads to targeting of the VDR?
Yeah, so that is a missing link. We have to figure that out. But I think the coating with vitamin D is changing the corona, and then that is trafficking it to the pancreas where it's interacting with the VDR.
Yeah, that's also exactly what I thought.
Yeah, so there is a missing link which we need to figure out, and we are excited about it, and we'll hopefully find something in the future.
And to your basically you choose then C12-200 ionizable lipid in your final pancreas formulations. So it would not work with, for example, D-lin MC3 or other ionizable lipids, right?
Yes, so this is through screening. I think yes, it didn't. It didn't. So
And how do you explain this?
corona. So it's like, yeah, I think it's surface decoration. It kind of blows my mind. I don't know. So what I feel like it is a combination of the decoration. So maybe these different ionizable lipid is decorating it differently and not making it accessible to the same corona. But we are, so these are all the questions we are interested in, and we will keep on working on this so that we can answer next time.
Yeah, it's really interesting, and would love to also have answers to these questions. Um, if it's okay, I would have one more.
Yes, please.
Do you think it could also work for targeting tumor tissue in the pancreas? And did
Yes.
you already perform experiments on this?
So right now I have not, but we are working on it, like to get there, because you need to have an orthotropic tumor. It's hard to get that right. So, uh, so that is in works. So checking in the orthotropic tumor and how it targets those tumor cells, number one, and then modulate the nanoparticles accordingly. But we do have some tumor targeting material which we also have to test, so we'll see.
Thank you so much. Yeah, it's highly interesting, very interesting papers, really nice work, and I'm following this, or we are following this here, so we're
Thank you.
excited what's coming.
Very good. This sounds like a developing collaboration right here. Excellent. Well, yeah, I mean, hats off again, Chandra. This has really been great, a great series of questions, a great start for your early career, and recognized on a number of fronts. And we were really excited to see what happens next with you, with the laboratory as you finesse some of these findings and continue to push the work forward. I can't see you anymore.
Oh,
There you are. Okay, great.
yeah.
So thank you again, and we'll look forward to speaking with you again, and have a great rest of your day.
Yeah, thank you for the invite. This was exciting. Thank you.
Great.